Am I audible and is my screen visible? Okay. Good afternoon, everyone. Welcome to the week 9 interactive session for the course Classics in Neuroscience. I am Ramya Shekhar. I am a PMRFT for this course. So, in this week's lecture videos, uh, the topics that were discussed were primarily regarding the Nobel Prize winning works of Santiago Ramon y Cajal in 1906 and those of Robert Barney in 1914. Let me first give you a brief introduction about Santiago Ramon y Cajal and his extremely pioneering and important contributions to the field of neuroscience and why he is regarded as the father or of modern neuroscience. So, who was Santiago Ramon y Cajal? So, Santiago Ramon y Cajal was a, a Spanish scientist who came from extremely humble origins. So, he was born in a small village, which constituted probably of around 200 or so residents in Spain, and his father was a barber surgeon. So, as a boy, Santiago Ramon y Cajal was he was not very good at studies because at that time the way of studying was that basically the child would be punished or beaten and made to learn by rote, and Cajal was not someone who was able to learn that way. So he actually apparently left school, but his father made him join back, and somehow he pushed through and finished his school studies. And as a child, he was also an apprentice for a barber and then for a cobbler. And he was extremely interested in becoming an artist. So he was very good at drawing and he really wanted to become an artist. But then again, his father, who was himself a surgeon, wanted Kahal to study medicine. So, having been asked by his father to study medicine, Kahal ultimately went on to finish his studies in medicine, and he he also became a military doctor so for a while, and he had to go on an expedition to Cuba for some during the war. And when he came back from that, he got malaria and tuberculosis, and he was extremely weak. I mean, during his expedition, he contracted these diseases, and he returned back in an extremely weak state. And then he tried to. Uh, Basically enroll in university as a professor, but it was extremely difficult at that time. Apparently, he failed his. Just a minute, yeah, yeah. So that I mean, he describes his um, school life as La letra con sangre entra, meaning that knowledge comes with blood, or basically that if you don't hit the child, then the child is getting spoiled. So, this is an idiom in Spanish, which means that basically, if you don't beat a child or punish a child, you are spoiling the child. So, and he described that, he describes that this is how his his school life was. And then later, when he wanted to uh, join for a position in university. He failed. So at that time there was this competitive exam called the opposition, and he failed that twice. So it was an extremely grueling competitive exam for being appointed in a university position, and he failed it two times because that required that you remember, like as in you have very good verbal memory. But Kahal was not someone who remembered things by rote. He was someone who wanted to understand things. So that's why he ended up failing two times. But finally, he passed on for one at the University of Valencia. And interestingly, his drawing that he made for the exam was so wonderful that apparently one of the judges even took the blackboard home. So that is how brilliant Kahal was in his draughtsmanship, basically, in his ability to draw. So, yeah, so this is Ramoni Cajal in his laboratory in Valencia in his early 30s. And as you can see that he's surrounded by microscopes. So at that time, from whatever savings that he had from his time in the war, he used every single money of that to buy a microscope which had a magnification of about 800 times. So that was he considered his microscope as his fifth child, apparently. Like he was so he used to spend days and nights and weekdays and weekends on making these drawings. So can you imagine, he used to keep he had to look in the microscope and he had to draw that. And these were the kind of intricate drawings that Kahal made. You can even see the dendritic spines and how the elaborate structures that Kahal has drawn by hand. So that he was extremely brilliant in terms of his uh, drawing abilities, and he was also an extremely wonderful researcher. So, yeah, and these are some of his other drawings. As you can see that he has drawn the pyramidal neuron of the cerebral cortex. And again, you can see the extent of detail that he has projected in these drawings. This one is the cut nerve outside the spinal cord. Again, drawings by Kahal, extremely intricate, extremely detailed. And he was someone who he was very patient. He was very rigorous with his experiments. He used to keep repeating his experiments in different systems, in different models, in different organisms, and he used to confirm his findings by repetition, and he used to very rigorously optimize his methods, and he was someone who had very high standards for experimentation. This is an image of glial cells in the mouse spinal cord. Again, another very wonderful drawing, as you can see. Sorry. These are glial cells in the cerebral cortex of a child. This is Kahal. He had, when he became older, he became completely bald. He's surrounded by several of his students, and he was always in lab, always researching. Even in his dreams, he used to think about. He even documented his dreams. He kept records of his dreams. He wanted to understand the neurobiology of dreams, the neurobiology of sleep. This is Kahal with his wife, who also helped him with his work. So she was, although she was relatively uneducated and an orphan, but they had a good married life, and she also helped him in the laboratory. In addition to managing the whole family as well as the joint finances, uh, Kahal's wife played a very important role in. So Kahal's wife also played a very important role. She was kind of his laboratory assistant, you can say, a laboratory technician. She helped him in several aspects of the laboratory work. She also managed the house and the family, and enabled Kahal to go for his conference in Berlin. So it is important to understand that uh, any scientific endeavor requires a lot of patience, a lot of support from your family. It requires a lot of hard work and extreme determination to keep doing things over and over and over again until you actually manage to reach the level of rigor required. Yeah, so we discussed about how he failed the oppositions two times and had finally managed to pass only in the third attempt. And why is he considered the father of modern neuroscience? Because he was the one who actually discovered that neurons are separate, independent entities and they are not a reticular network. So he was the one who came up with the neuron doctrine. And in addition to the neuron doctrine, of course, he had made several other important contributions. We discussed about his ideas about sleep, his ideas about dreams, his extremely insightful thoughts regarding various aspects of neuroscience. And also, so how did this start? So, how did Kahal get to, I mean, how did he realize that neurons are independent entities? So, what happened is that while Kahal, so around 1887 ish, Kahal, through an acquaintance, he came to know about the stain that was developed by Camillo Golgi. So Camillo Golgi was an Italian Italian who had developed the Golgi staining method, which is basically a silver staining method. And through an acquaintance, Ramoni Kahal got to know about the existence of this method. And but the problem was that that method that Golgi had developed, it was not very reliable. So sometimes it used to stain the tissue all right, sometimes it was not very reliable. Almost nothing was visible, and people had kind of almost given up on trying to use the Golgi stain. But when Kahal got to know about the existence of this method, he spent several months trying to perfect it. So there were only two components for that staining method, but he had to optimize the exact concentrations, he optimized the durations, he optimized the different kinds of he tried different kind of regimes. He tried to do the stain more than one time, two times, three times, and he tried to use different tissues. A lot of optimization went into that, and a lot of patience was of course required. And that way Kahal actually improved the original Golgi stain and then used that to study almost every kind of tissue, every kind of nervous tissue imaginable. So he was spinal cord, he tried to study the cerebellum, the cortex, almost every different kind of nervous tissue. He even tried to look at embryos. So he decided that because the adult brain contains white matter which has myelin, and myelin will prevent the impregnation of the stain. So he thought, okay, let me try to see what happens in embryos, because in embryos, because it is less complex than the adult, the relative complexities and the relative problems that he was facing with the staining will also be reduced. And because there will be less myelin in the embryonic stage, it will be much easier to stain the tissue. So he even tried to use the embryos. He also tried to check it in different kinds of tissues, different organisms, and finally, so he made extremely elaborate drawings of whatever he was seeing under the microscope. And all of these helped him to realize that the structures that he was seeing, they were independent entities that were in touch, but they were not touching each other. Basically, he meant that these are independent entities. They are not a reticulum that Golgi and others were proposing. It was not a net. They were independent entities which were in communicating with each other. And so when other people were taking a look at the stain samples, they were they somehow were not able, so they believed that it was a tangled network, and the whole nervous system is a reticulum which has a single identity. Whereas Kahal and several others realized that this was not the case, that there were actually breaks, or there were actually like now the ones that we call as synapses, they were actually breaks. They were actually places where there is no staining. As in, for example, here you can see there is a break. It's not a reticulum. So this was because Kahal was able to uh, very carefully visualize the nervous system using his improved method, and because he also tried to do this in several kinds of tissues. So he himself was not initially, he was not sure whether he was going in the right direction, but he kept repeating his experiments, and he used different tissues, and he kept getting the same answer, that these are independent entities. So this is how Kahal came to uh, like improve the Golgi staining method and make it his own, and come up with these extremely brilliant observations, and use that to propose the neuron doctrine. Funnily, the term neuron, although it was Kahal who discovered that they were independent entities, the term neuron was not attributed to Kahal. I mean, he was not the one who came up with the term. The term was coined by Waldeyer, who had also coined the term chromosome. And unfortunately, people at that time misunderstood that it was Waldeyer who had discovered the neuron. Rather, it was Kahal, and Kahal felt extremely bad about this, and he was reminded again that he was practicing science, or doing science in a country where it was very difficult for him because of all the administrative problems, the relative, the basically the environment in which he was like, struggling a lot to do science. And apparently he had remarked that in Spain, to do science means to cry. So it was a very difficult period, difficult time for Cajal in Spain to carry out the science that he was doing. And Spain was also not in the same league as other nations at that time in terms of the scientific endeavors. So France, Germany, Britain, Great Britain, all of these were considered to be very good, excellent centers for science. Spain was nowhere in the picture. So, like not initially, people did not even realize or know about what Kahal was doing. It was only when Kolliker, who when Kolliker saw, so Kolliker at that time was a very big, very well recognized stalwart in the field. So when Kolliker saw Kahal's slides, and when Kolliker remarked that Kolliker was extremely enchanted by the work that Kahal was doing, and he wanted the world to know about him. And slowly that Collicum basically helped Cajal become known to other scientists, for example, in Germany. And then Cajal's work got translated to German, and other people got to know about his work. And slowly, that's how Cajal got to like was it was realized that this was the kind of work that Cajal was doing. So Kolliker played a very important role. But there also, it was Kahal had to actually like really force or beg Kolliker to take a look at his work and see what he was trying to do, and that is how it happened. So Kahal faced a lot of difficulties to carry out his science and tell the world about it, because he came from a country where scientific endeavors were not really at the same level as other parts of the world at that time. So, who is considered the father of modern neuroscience? It is Santiago Ramon y Cajal, who won the Nobel Prize in 1906 for in physiology or medicine for his groundbreaking uh, discovery about the structure of the nervous system or the structure of neurons. So, what was Santiago Ramon y Cajal's major contribution to neuroscience? It was the discovery of the neuron doctrine. And like we know, he was against the reticular theory. He did not believe the reticular theory. And the reticular theory, one of the key proponents of the reticular theory was Camillo Golgi. So the one who actually developed the Golgi method that Cajal later refined and used. That was Camillo Golgi, but Cajal and Golgi were rivals, so to say. And what happened was, during the Nobel Prize lecture, first Golgi gave the lecture, followed by Cajal. And in his, so they both shared the Nobel Prize that year, ironically. And during his lecture, Golgi strongly defended his reticular theory, and it kind of like dismissed the neuron doctrine of Cajal. And later, when Cajal gave his lecture, he strongly advocated for the neuron doctrine. So it was a very, unpleasant, you might say. So, yeah, has written in his later. Cajal has written that what an irony of fate it is that the two of us were joined like Siamese twins for winning this Nobel Prize, and both of us are like completely opposite in our what we are proposing. And he felt quite bad about how Camillo Golgi had behaved during his Nobel lecture. So, and Kahal was also of the opinion that, I mean, one should, I mean, if some new hypothesis is coming up and that is true, I mean, or that there is more evidence to support that, then one should gracefully uh, let go of one's uh, dogmas and egos and accept the truth rather than staunchly just defend one's views just because they were the one who came up with it. Also, I would strongly recommend you to if you are a researcher or if you have any interest in research or any interest in becoming an investigator or a PhD student or something, I would strongly recommend you to at least go through Kahal's book called Advice for a Young Investigator, if I'm not wrong. That's the title of the book. It is a very insightful book in which he has discussed a lot of, he has given a lot of very good advice that even till date is applicable to young investigators, telling them about the trials and tribulations they might face as they start research and how they should deal with it and what kind of worldview and what kind of views they should inculcate in their research or in their pursuit of science. Yeah. So, what technique did Camillo Golgi develop to visualize neurons? Camillo Golgi developed a technique which he initially termed as La Raisonnera. I'm sorry for the incorrect pronunciation. So, basically, the black reaction. So this was what Camillo Golgi had developed. Like I said, Camillo Golgi was an Italian. He Although he developed this staining technique, it was not very standard or rigorous, and it worked sometimes, it did not work in other times, and it was initially it laid on like kind of dormant and was no one was interested in actually pursuing this. It was much later when actually Kahal started using this and modifying it that it actually like became revived, to be so to say. So, what technique did Golgi develop to visualize neurons? It was the black reaction. And why was it called black reaction? So, here you can see this is a human neocortical pyramidal neuron. And here you can see that, so the deposition of the Golgi precipitate has given this, has made the tissue basically black, right? And so what happens is basically it's a silver staining technique. So I will come to the exact procedure and so on in a minute. So it was a silver staining technique, and sorry, yeah, it was a silver staining technique in which uh, because it gets the silver gets deposited in the cell bodies and dendrites, so the tissue acquires a dark black structure, um, like contrast. So because of that, it was called the black reaction. And Golgi did not develop the double impregnation method. The double impregnation method was actually developed by Kahal. So, what was the difference between the Golgi method and the Kahal method was basically, like I said, Kahal optimized the concentrations and the durations and so on. So, basically, there were only two components. One was potassium dichromate, and the other one was silver nitrate. So when what happens is that in the Golgi technique, basically there were the precipitate was basically the microcrystals of silver chromate. So, but the problem was that it was not very, like I said, it was not very reliable. So, what Kahal did was that, in addition to changing the durations and changing the concentrations, Kahal also stained it with the same. He also performed the staining more number of times. So if you do it two times, so that was the double impregnation method. So basically you're impregnating the tissue two times with the same stain. Or he even tried it for more times, like three and so on. And it took Kahal a lot of time to optimize and basically come up with the double impregnation method so that he could visualize the neurons with a great level of detail. And it also allowed even the more elusive cells and of the nervous system which were initially not being which were not visualized by Golgi's technique, even those were stained by Kahal's improved technique. So yeah. Electron microscopy, of course not. Electron microscopy came up in the 1950s, whereas these techniques were much older. These were in the 19th century. Mid to late 19th century is when Golgi's black reaction and Cahal's double impregnation method came into the picture. So Cahal's double impregnation method was probably around 1887. Black reaction, I'm not exactly sure about which year, but it was much older. So I think it was in the probably in the 1850s or so, if I'm not wrong. I'll have to. So yeah. Neuroplasticity staining. So at that time, there was not even an idea about what about neuroplasticity as such. Although Kahal did have some really interesting theories and ideas which uh, anticipated neuroplasticity. So Kahal was actually much ahead of his time in terms of his ideas and theories, and several things that Kahal has made a note of, the several ideas that he came up with were actually much relevant and continue to be relevant even now. Sorry. So far, is it clear? Is there any doubt that you guys have? Okay. Yeah, so this is a portrait of Camillo Golgi. And by the way, remember the reason why Kahal had to make those extremely intricate drawings by hand, because at that time there was no available way of photographing microscopic images. So it had to be done by hand. And not just Kahal, but even several other histologists at that time they had to be good, had to draw their observations. That was the only way in by which they could document their observations. Yeah, so coming back to the black reaction developed by Camillo Golgi, what was the procedure? So the procedure was basically like this. So the freshly removed nervous tissue was first hardened and then fixed in potassium dichromate for several days and then left successively for the variable periods of time in silver nitrate. Then the blocks were dehydrated and then cut. Just a minute. Yeah, so then the blocks were first dehydrated and then they were cut without any embedding. And then the sections which were extremely thick were cleared in turpentine and attached to slides and covered with gum damer and without using any cover slips. And then to observe the sections from both sides, Golgi used to mount them on a cover slip, which he positioned over the aperture of a hollowed out wooden slide. So here you can see what when you look at the procedure itself, you realize that the time duration for which the tissue is left was variable, and the time duration in which the tissue was left in silver nitrate was also variable, and the concentrations that were used of these different solutions were also variable, and they were dealing with extremely thick sections of tissue, and they were not able to perform any embedding, and in many cases they were not using cover slips for their experiments. So lots of different variables, and therefore it was very unreliable to perform this technique. And so, what was the mechanism by which this Golgi staining worked? Basically, the nervous tissue, which was fixed by aldehyde, was impregnated. Impregnated basically means it was stained. So, it was penetrated basically with potassium dichromate and silver nitrate. And that resulted in the cells being filled by microcrystallization, basically extremely small crystals of silver chromate. So there was basically a reaction between silver nitrate and potassium dichromate, which resulted in the formation of microcrystals or extremely small crystals of silver chromate. So it was a silver staining technique, and this microcrystals of silver chromate is what gave rise to the black color or the black precipitate that was observed in the and that allowed them to visualize the nervous system, the cells of the nervous system basically. But like I said, Golgi technique also was not very good in terms of not many cells were not visualized, many parts of the cells were not clearly visualized, and so on. So when Kahal first, like I said, Kahal got to know about the existence of such a Golgi staining method from an acquaintance, right? So when Kahal saw this, he was extremely fascinated by this technique, and he decided that he will try to adopt it and modify it. So when Kahal saw, what was Kahal's opinion of this method? So this is quoted from what Kahal has written. I expressed the surprise which I experienced upon seeing with my own eyes the wonderful revelatory powers of the chrome silver reaction and the absence of any excitement in the scientific world aroused by its discovery. So Kahal was extremely fascinated by this method, and he was extremely also surprised that why the world has not taken notice about such a revolutionary method. This is a drawing by Camille Golgi himself. Again, like I said, at that time, you had to you had to draw. You had to draw whatever you're looking at under the microscope, and when you published, it was the drawings that you actually published of your observations. And because at that time there was no way to document what you were looking at under the microscope, so all the histologists of that time had to draw their observations and publish them. So this is one of Golgi's own drawings in which he has used his black reaction, basically his Golgi staining technique, and this shows a multipolar neuron. So what is a multipolar neuron? It has basically several processes, as you can see. This is the cell body, and these are all the different processes that are coming out of the multipolar neuron. So he has used his black reaction to impregnate the tissue, and this is his observation of a multipolar neuron from the spinal cord of a mammalian fetus. Yeah, so next question. What is the Barony test primarily used for? So the Barony test was actually a test that was developed by Robert Barony. So Robert Barony was an Austrian of Austrian-Hungarian origin, and he came up with this test to check whether the vestibular system is fine. So, in many patients who experience uh, dizziness or motion sickness, or they have problems with balance, they are they feel very dizzy and nauseous and vomit because of a lack of orientation or a lack of balance. For example, in case of vertigo, in those cases, uh, before Robert Barony came, described the mechanism, it was understood that several other scientists had performed work which made them realize that the vestibular system is somehow involved. So they understood that the vestibular system is basically a system that is present in the inner ear. So it was realized that the inner ear has some role in maintaining the in telling the brain about the balance of the person. So several other researchers had understood that there is the vestibular system has some role, but they did not know what it was. They did not realize how it works. So although they had realized that the vestibular system is involved, they had no idea about how it worked. But Robert Barony, who again had studied medicine, he was the first person who studied and described how this vestibular system works, and what is the mechanism, and why in certain patients and certain individuals with conditions like vertigo, or in individuals who have suffered some kind of damage to the vestibular system or to the brain stem, there are these problems with balance. So he developed this test called the Barony test to assess vestibular function. So, these are the semicircular canals of the vestibular system, and this is the atricle. So the canals are responsible for detecting the rotational motion, whereas the atricle and there is another part. I'm currently forgetting that. So those are responsible for detecting the linear motion. I think it's called succulus. Please go back and check that. But so there are two. Those two regions are responsible for detecting the linear motion, whereas these semicircular canals are responsible for detecting the circular motion or the rotational motion, basically. Yeah. So Robert Barony developed this Barony test to assess vestibular function. So how does that work? I'll come to that in a little while. I have an image in the presentation which will explain how the Barony test works. So Barony received the Nobel Prize for his work, and he was a prisoner of war at that time in Russia, and he had to, like, the Prince of Sweden had to specifically request for his release so that he could accept the Nobel Prize. He accepted the Nobel Prize almost 2 years after it was announced in his name. So he got it. He received it in he was supposed to receive it in 1914, but since he was a prisoner of war in Russia, it was only upon his release, which was catalyzed by the Swedish prince, that he received it in 1916. And then he settled in Uppsala, and he continued his work on the vestibular system over there. And later, he also made several other important contributions to the field. So, what was the key finding of Robert Barony that led to his Nobel Prize? He discovered the caloric response. So there is a caloric response, the caloric reflex test also that is conducted by ENT doctors. So he was the one who discovered this caloric response. And what is that basically? So he discovered that in if you like pump with a syringe uh, hot or say warm or cold water into the ear of a patient. Basically, if you pump the water that towards the inner ear of the patient, what happens is that there are ocular reflexes that take place. So um, the eyes basically move involuntarily from one side to the other. So he discovered that if you the cold water induces one kind of response, whereas warm water induces a different kind of response. And he understood, or he realized, that it is because of the changes in convection in the ear that is responsible for the dizziness that is experienced by the patients. So the endolymphal fluid that is present in the inner ear, that responds to changes in convection. So if the water that is pumped is warmer, then that will cause a different response. If the water that is pumped is colder, that will cause a different response. So the involuntary movements of the eyes will change. And remember, the both the ear and the eyes report the state of the individual back to the brain. So if the eyes are conveying something and the brain and the ear is conveying a different thing, then of course there will be a problem. So, for example, in case of say motion sickness, if a person is sitting in a driving car and they are trying to read a book, for example. So the eyes are looking at a stationary object, whereas the ear is telling the brain that the individual is actually in motion. So therefore, there is a problem because the brain thinks the brain is not able to understand whether you're in motion or whether you're stationary. So you experience motion sickness. And in case of vertigo, what happens is that vertigo is again another is also caused by basically problems in the vestibular system. So what happens is that there are free floating autoconia or calcium crystals that uh, come out of the urethra and into the canal. So, because of these crystals, the individual experiences vertigo. So, Barany also discovered a form of vertigo called I, th- I think I have it somewhere. So he also discovered a form of vertigo, and he also recommended the benign paroxysmal positional vertigo, which is characterized by the presence of these calcium crystals in the semicircular canals, which get displaced from the ventricle and they enter these canals. And that is the cause of this vertigo. And he was the one who studied this benign paroxysmal positional vertigo or the BPPV. And so there is this maneuver called the Epley maneuver, which uh, can help individuals to basically overcome their vertigo because it basically these crystals are sent back to the ventricle. So if there is debris in the posterior semicircular canal, that causes the vertigo, and by this Epley maneuver, the uh, debris is basically diverted to the ventricle. So the patient experiences relief. Yeah, so coming back to Robert Barany's finding, he discovered the caloric response. So, caloric response is basically it says that the different temperatures in the inner ear can cause the flow of the difference in the flow of the endolymphal fluid, and because of that the person will experience changes in their sensitivity to their sense of balance. So the hair cells which are present in the inner ear, their sensitivity will differ. So if the water is warmer than say 44 degrees Celsius, then it will induce one kind of response. And if the water is say colder than 30 degrees Celsius, that will induce another kind of eye response. Next question. What is the law of dynamic polarization proposed by Cahal? So, like we discussed, Cahal was the one who came up with the neuron doctrine. or the fact that neurons are independent entities which communicate with each other, but they are they do not form a reticular network. So that was what Kahal had proposed. Then, through his very extremely careful observations, he also saw that the dendrites. So he, like I said, he studied many different systems. He studied the retina. He studied other sensory systems. So he saw that in all of these cases, the dendrites are position towards the outer side, towards the outer world. So, or the basically towards sensing the stimulus. Then comes the cell body, and followed by axons. And he saw this in different systems. So, because of that, and because like he of his other ideas and observations, he came up with this law of dynamic polarization, in which he basically proposed that there is a polarized flow of information within neurons. And this polarized flow of information basically happens from dendrites to the cell body to the axons. So he said that the nerve impulses travel in one direction within neurons because of several observations, including the fact that the dendrites face the outer, in the sensory systems, he saw that the dendrites face the outer world, followed by the cell body, and then by the axons. So, although, I mean, Kahal came up with this law, now we know that, so now there are refinements, and we know that this is not always the case. Now we know that there is also retrograde, and there is also axonal and dendrodendritic flow of information. It is not just one directional. It is not unidirectional. It is, we know that there are other directions also by which information flows within the nervous system. So this is not, I mean, it is, while Kahal proposed it at that time, now there are exceptions, because we know that dendrites are not just passive elements, they have active properties, and they can also conduct impulses. So, we know that dendrites, I mean, there are dendrodendritic signal impulse transmission also, which happens, for example. So that was the law of dynamic polarization that Kahal had proposed, that information travels only in one direction from dendrites to cell bodies to axons. Next question. What condition did Barani's research on the vestibular system help explain? We have already discussed this. We discussed that Barani, because of his work on understanding how the semicircular canals work, basically by pumping using a syringe, either very warm or cold water, he realized that the so because of the because of the changes in the convection in the inner ear, what happens is that the semicircular canal, the endo lymphal fluid that flows through these canals, the flow of that is altered. And because of that, what happens is that individuals experience, there are certain individuals who experience more, who become more sensitive, or they are excessively sensitive to changes. And he also showed that, he also studied the deposition of these canalith particles or crystals in the semicircular canals, which causes the discomfort that is experienced by the person having the benign paroxysmal positional vertigo. And we also discussed the Epley maneuver. I would urge you to go and check it out. You might, I'm quite sure there are YouTube videos and other such resources which you can take a look at about how this Epley maneuver is performed as a treatment for BPPV, in which basically the canalith particles or the crystals which get deposited in the posterior. So there are three different sorry so there are three in the semicircular canals are basically three different rings, right? The posterior canal, the lateral canal, and the superior canal. So these calcium crystals get deposited in the posterior canal, and because of that, the individual experiences vertigo. So this deposition of the debris in the posterior semicircular canal causes the symptoms of the syndrome. And when the person performs this maneuver, what happens is that the canal, so initially the posterior canal gets inverted, and the debris starts to settle, and because of that there is nystagmus. So nystagmus is basically what I was describing earlier, the involuntary eye movements that happen. So rapid involuntary movements of the eye are referred to as nystagmus. Yeah. So through this maneuver, what happens is that finally the debris gets deposited in the artericle. So it leaves the posterior semicircular canal and goes to the artericle, and because of that, the person experiencing vertigo gets relief. Interestingly, Barany also showed that in individuals experiencing vertigo, with so initially the person might experience discomfort. Say if say one, so there are two vestibular systems, right? One in each ear. So initially, so say if someone has a damage in one of the vestibular systems, and they are experiencing vertigo. But Barney also showed that with age, there is vestibular compensation. Basically, it means that with age, they are somehow they are able to the symptoms of the syndrome decrease, or the brain is somehow able to adapt or become more basically one of the vestibular systems is given more priority over the other. So the one that is normal is given, the brain gives more like receives information from that and processes that, and because of that, the symptoms of the syndrome might stabilize or decrease with age. So that is basically an example of an example of neuroplasticity in the vestibular system. And this is also something that Barany showed. And Barany, like I said, initially he was a prisoner of war, and then after that, when he was released, he settled in Uppsala, and he continued his research over there, and he made these other findings. He continued to work on the vestibular system. His work also helped in uh, other aspects, such as understanding motion sickness, understanding. So the brain stem is the one that receives the information from this vestibular system, and if there is any damage to the brain stem, then also the person will experience problems in balance. And Barney's work therefore had a very long lasting impact, because it helped not just these kind of patients who had issues with balance, but it also helped us our improve our understanding of the so what happens if an astronaut goes to space, because Barney showed that there is a because of this mechanism and the fact that gravity plays a role in the vestibular system, and because in space there is no gravity or very less gravity, what can happen to an astronaut. So the disorientation they will face, the dizziness that they will face, and how treatments or how trainings can be provided to them with regard to these with regard to this. So because of the caloric response that Barney discovered, and his contributions to understanding the vestibular system much better. It helped in patients with different kinds of diseases. It helped in for astronauts. It helped people experiencing motion sickness. It helped military personnel who were the military the air pilots and the fighter pilots, and even the naval officers and the sailors. So it had a very long lasting effect. And also Barney's research helped in the development of medications or medicines for motion sickness. Yeah, so but Barney was not the first person to investigate the phenomenon of vertigo. It was actually Perkinjay, who has contributed. So this is Perkinjay, who was a Czech physiologist. So he was someone who investigated the phenomenon of vertigo, and he came up with this law of vertigo. So what did he come up with? He came up with the law with an observation that the apparent motion perceived when stopping the head after rotation changes from horizontal to vertical if the head is inclined laterally. So, based on how the head is inclined, if the head is inclined like this, or if the head is inclined straight, versus if the head is inclined in other positions, then the way the motion is perceived, or the apparent motion is perceived, is different. So the way the head is oriented determines the motion that you perceive after the rotation is stopped. So if an individual is placed uh, with his head in a particular orientation and is rotated, and then the rotation is stopped, then the motion that individual perceives after the rotation is stopped, so when the person has become stationary again, at that time the motion that they perceive depends upon how the head was oriented. So if the head was inclined laterally, then the motion that is perceived changes from horizontal to vertical. So basically, he came up with this law, which, like this law of vertigo, and like we now know, this law is important to many individuals, even to pilots, even for astronauts, for sailors, and so on. So for pilots, because when they during uh, say pilots, as well as in the military, the air force, for example, in both conditions, what happens is that if they move the head down, or if they move the head laterally, then that can induce uncontrollable vertigo if there is no external reference. So, this was Purkinje's law of vertigo. And the work of Purkinje came before that of Barony. So, like I said, Barony was not the first to study the phenomenon of vertigo. There were others. So, Purkinje was one of them who studied the law, who came up with the law of vertigo. Next question. Who investigated the functions of pigeon brain? So, Pierre Florence. So, this is an image or a drawing or a portrait of image Pierre Florence, who was a French physiologist, who not just he is regarded as the father of experimental brain science, and he also did some pioneering work in terms of anesthesia. So, Pierre Florence investigated the functions of pigeon brains in the context of balance. So, at the time, what was popular is that, like I said, so even in case of Purkinje, you saw that the uh, the the creature is made to rotate. So at that time there were these these devices that were these contraptions, or these uh, devices that were being made, which allowed the sample, the basically the animal, or the individual, or the human, to rotate. So by rotating, basically like a rotating chair or a rotating cage, or basically different kinds of rotating uh, devices were used to investigate the effects on the uh, person's sense of balance and orientation. So, Pierre Florence used pigeon brains to try to understand the role of different regions of the pigeon's brains on different aspects, for example, in terms of learning and memory, in terms of balance, and so on. So, is that clear, or do you have any doubts? Okay, thank you. So, In that case, I will close today's meeting because uh, I have discussed the previous year assignment questions, and I hope I have given you a good overview about the life and works of Santiago Ramon y Cajal and Robert Balmy, and the kind of trials and tribulations that they faced in carrying out their science, the discoveries that they made, the contributions that the impact of their contributions. Okay, the impact of their contributions in different fields and how they influenced several researchers across the years. So, the Epley maneuver is basically the patient or the individual is made to lie down and their head is tilted first in one direction to the right. So what basically happens is that initially the patient is having these crystals or these debris in their posterior semicircular canal, right, and that is causing the discomfort. So when the patient is made to lie down, their head is tilted to the uh, right, and that causes the debris to settle uh, because the posterior semicircular canal gets inverted. And because of this settling of the debris, there is nystagmus. So basically, there's involuntary movements of the eyes. So now when the patient's head is tilted to one side, the debris is settling. Then the patient's head is tilted to the opposite side and it is held for 30 seconds. Then the patient is made to get inverted, like the patient inverts his body like this and on one shoulder, basically, on one side, basically. Again, hold for 30 seconds. So the debris comes out of the posterior canal and enters the article. So, like you can see, this is the anterior canal, this is the posterior canal, this is the lateral canal, and this is the article. So, these anterior, posterior, and the lateral canals are the semicircular canals, whereas which are responsible for the detection of the rotational motion. And the article plays a role in detecting the linear motion. So, when the when this maneuver basically when this maneuver is performed, basically the debris that first got settled uh, in the first few steps, that enters the article from the posterior canal. So now that the debris has entered got deposited in the article instead, the patient experiences relief. So this is called the Epley maneuver, which is used as a treatment method for benign paroxysmal positional vertigo. Is that clear? No problem, Amrita. Oh, interestingly, regarding Santiago Ramon y Cajal, Cajal became famous, he became so famous that he was offered the position of an education minister in Spain. But Cajal refused that position, and he decided to concentrate completely on his academic endeavors. Although he did help in drafting some uh, advisory, uh, his own advisory recommendations for education. And Kahal was also someone who uh, he was always in touch with nature. He always wanted to understand nature. He was always very observant. And as a child, he used to collect bird eggs, and later, as an adult, he carried out his experiments on eggs. He was someone who believed that there is no Sunday in nature. So he used to say, apparently, that is are there Sundays in nature? No, right? So he was someone who devoted almost all his time to research. He even he was interested in understanding hallucinations. He was interested in understanding the biology of delusions, of mania, of like he even tried to understand. So he initially he thought he wanted to also understand about spirits and so on, but later he said that, I mean, he gave up on that because he found that one medium was actually a fraud. And then he said that he got very um, I mean, his belief regarding that faded, and he stopped that research on that. But and he even used hypnosis. Apparently, he also he even tried to understand and use hypnosis, and apparently he used to carry out hypnosis on patients, and that used to help them in certain cases. He even carried out hypnosis on his wife during childbirth to uh, provide her relief, apparently. So Kahal was a man of many pursuits. He made many different contributions. He was the one who also came up with the he one who was the first to observe and describe the axonal growth cone. So, which is he described it as a battering ram. So that goes through a lot of traffic to make contact with its target. So he was the first one to observe the axonal growth cone, and he came up with the term to describe uh, these uh, um, these parts of axons which are responsible for the making contact between of the axon of a neuron with its target. So, Kahal has made extremely valuable contributions to the field, and therefore he is rightly regarded as the the father of modern neuroscience. And apparently, so not apparently, there was a Cajal Institute that was established in Spain in his honor. Uh, when he was alive. Not, I mean, generally these kind of institutes for on which are in honor of an individual are generally it has been generally it is established after an individual's death, right? But in this case, in Kahal's honor, the Kahal Institute was established when he was alive. And so Kahal died in 1934, and he was someone who had like devoted all of his efforts to understanding the nervous system. So initially, before 1887, he was working on several other aspects of human biology as well. But in 1887, he decided to abandon them all in favor of only trying to understand the nervous system. So he had this single-minded goal of understanding the nervous system, and he published prolifically. He published several monographs and several articles over a very short period of time. Extremely productive work and extremely rigorous. And he used to spend days and nights hunched over his microscope and carry out his research. Many of Kahal's students and disciples had to flee or exit or run away from Spain during Franco's regime. So, because like a few years after Cajal's death, so Spain was occupied by the Franco regime, and many of Cajal's students had to flee because uh, Franco and his regime basically did not allow, were not in favor of Cajal's neuron doctrine, and uh, they brought back the Golgi's reticular network ideas back, and they had, I mean, Kahal's disciples faced a lot of problems basically. And if Kahal himself were alive, he would have also not been, he might have also been persecuted. It is quite possible. Yeah, so that is Ramoni Kahal for you, who described his uh, life in Spain as a researcher as extremely difficult, and he lamented the lack of uh, scientific opportunities, and still he managed to make such extremely brilliant discoveries and came up with extremely brilliant ideas. And he that's why he was, and like I said, because he came from extremely humble origins to become what he became. So he was described as the peasant genius by Sir Charles Sherrington. Because, I mean, relative to the more, what do you say, more opportunities that people like Sir Charles Sherrington enjoyed in England, for example, Kahal did not have that. So, for example, Sir Charles Sherrington enjoyed a relatively better ambience and environment in Oxford, and because Britain at that time was doing fairly well in terms of the scientific endeavors that were being carried out, but that was not true for Spain. So Carl was the first Spanish national to win a Nobel Prize, and it was like, I mean, he tried a lot to reform the system in Spain, but unfortunately, it was not to happen. So that is all about Ramoni Kahal. I hope you had a good time learning about this great individual who gave us the uh, neuron doctrine and gave us our extremely beautiful drawings and which helped our understanding of the nervous system. And these drawings continue to remain classic works and are reproduced in almost every neuroscience textbook that you would see. And of course, let us remember the how he was supported by his wife. Apparently, his wife was regarded half of Kahal because of how much she supported his endeavors and helped him in his endeavors and managed the family as well as the finances and helped him in the laboratory and helped him get funded to go all the way to Berlin to present his work. And it is quite interesting that when he presented his work in Berlin, I mean, he found it very difficult to go there because there was very less funding that he had available. But he somehow managed to go to Berlin to present his work in Germany. And that is when I mean, that is when the world got to know about the work that he was doing, because initially his work, which he was publishing in Spanish, I mean, only when he interacted with Colliker and Colliker helped him get recognized. That is when his works were published in German and the world got to know about him. And Waldinger synthesized the work of Cahal, Golgi, and others, and came up with the term neuron and so on. Yeah, so with that, if there are no further questions, I hope you had a good time learning about Ramoni Kahal and Robert Barani and their important contributions to the field of neuroscience and how it impacted our understanding of neuroscience in the modern, I mean, till date, how we have been influenced by their work and how it helped us in developing imaging methods, developing treatments, understanding various aspects of nervous system structure and function, and so on. So, with that, I come to the end of the session. If you have any doubts or questions regarding the interaction session, whatever we discussed today, feel free to drop your questions in the discussion forum. I would be happy to respond to you. Thank you very much, everyone. Have a happy weekend. Thank you.